All right, we ready? Pretty ready? Mostly ready? All right, so we left off last time checking out the citric acid cycle. So we had finished glycolysis to make our pyruvate. We got to do this step to get to acetyl-CoA, and then we can enter the citric acid cycle. So let's take just a big, general, huge overview of the citric acid cycle and see what some features that make it unique are. So firstly, it is a circular pathway that oxidizes acetyl groups from a variety of sources. So we are specifically talking in this class going from glycolysis to citric acid cycle. However, you can also enter the citric acid cycle from fatty acid synthesis, and or the amino acid synthesis. So you can enter the citric acid cycle from a variety of different pathways: fatty acid, amino acid, or glycolysis. The citric acid cycle gets the cool name the hub of metabolism. So it's a hub, kind of like an airport's a hub, right? Lots of different airports feed into it, and then that gigantic airport feeds out to a lot of other cities. All right, its overall net reaction we're going to have 3 NAD pluses, 1 FAD, 1 GTP on inorganic phosphate, and an acetyl-CoA all enter the cycle at a variety of points. From that, we produce three NADHs and an FADH2, 1 energy unit, a GTP, a free coenzyme A, and 2 molecules of carbon dioxide. All right. In eukaryotic systems, all the enzymes of the citric acid cycle are located in the mitochondria. This is going to be my 1 minute of biology for today. Mitochondria. So all the substrates plus the NAD plus the GDP are either generated in the mitochondria from other cycles, or they're going to be transported in from the cytosol. So this is a system that takes place all within one area. And we'll talk about it some more in that what are the advantages of having all your enzymes and substrates and products all found in one area in terms of efficiency. Okay. One carbon dioxide is produced per 1 round of the cycle. That's not what I want to say. Scratch that off. That's not the right sentence. I want this. So the carbon atoms that are in are 2 molecules of carbon dioxide. The carbon atoms of the 2 molecules of carbon dioxide produced are not the same 2 carbons that started the cycle. So we're going to see some bonds breaking and bonds forming, and those 2 carbons that enter are not the same 2 carbons that make carbon dioxide. They go on to do other things. So looking at our net reaction, where we have NAD plus and FAD making NADH and FADH2, what sort of reactions would you expect to see a lot of in this cycle? Every time we see NAD and NADH, oxidation reduction. Yep, so we're going to see a lot of oxidation reduction in this cycle. So the oxidation of the acyl groups to 2 carbon dioxide molecules requires the transfer of 4 pairs of electrons. So we're going to see 3 NAD pluses overall plus 6 electrons and 6 protons make 3 NADHs. Then we're also going to see an FAD plus gain 2 electrons, become FADH2. Oh yeah, question, Richard.
What's after the 6 electrons?
6 protons. That's right, overall it's 4 pairs, 8 electrons, 4 pairs in different reactions. All right, so this is some big picture things, big pictures. So let's take a look at how do we actually enter the citric acid cycle through pyruvate dehydrogenase multi-enzyme complex. So looking back at our cycle, right, we made pyruvates, but we had to go from pyruvate to acetyl-CoA in order to enter the cycle. This is step 0 of the citric acid cycle, or whatever you want to call the step before the actual cycle starts. We're going to use pyruvate dehydrogenase, which is what we saw when we were talking about ethanol production. It's the same enzyme. It is a multi-enzyme complex. So multi-enzyme complexes mean it's a group of non-covalently associated enzymes that catalyze more than 2 really, sequential steps all at once. So pyruvate dehydrogenase has 3 enzymes in it that are all associated together. Let's take a look at how that works. Do I have that on the next page? No, okay. So, what is the advantage of using a multi-enzyme complex rather than 3 enzymes like we've done in glycolysis, where it goes from step 1 to step 2 to step 3? What would be the advantage? It should, in theory, be faster. So, enzymatic rates are limited by the frequency of collision. As you're imagining, right, in order to have the enzyme react with the substrate, they physically have to find each other, have a collision, do the chemistry, float away, and then do the next step. If we use a multi-enzyme complex, the distance traveled for substrate and enzyme is minimized. All right, so if we have all 3 enzymes in the same complex, the substrate becomes the product, which immediately is right next door to the next enzyme it's going to use. So that should speed up the reaction because it doesn't have to diffuse through space to find the next step. It also minimizes the opportunity for other reactions to happen. Right, it should limit the side products or the intermediate being used by something other than these complexes. It should streamline the whole process. So, what are the enzymes that are in this pyruvate dehydrogenase? These are the 3 enzymes that are contained within pyruvate dehydrogenase. Pyruvate dehydrogenase is one of them, which is why it's called that. There's also a dihydrolipoyl transacetylase and a dihydrolipoyl transacetylase and dehydrogenase. How this molecule all works together. So if you can imagine, here's a little cube, right? So you have your cube, which has 8 faces. If I put the E2 on every edge of the cube, and I put 2 on every edge, you get 24 E2 proteins. One layer up from that, on the faces, we'll put the E1, Sorry, E3. I color coded that wrong. That should be E3. The 12 E3 proteins are on the 6 faces, and the 24 E1s are on the edges. There we go. So as we put it all together, on the inside are the blues. Then on the outside are our greens and reds. So all together, this is a 60 subunit complex with multiples of every enzyme in it.
So you see like A
A is inside B.
and B.
No, C is putting it together. So A is this, 24 E2s on the corners of a cube. Then B is the E1s and E3s. Then C combines A and B. The same figure out your book if you want to see it in color. But this cube is inside this cube to make 60 subunits all together. So it should be a very efficient enzyme, right? Because you have 60 subunits, which are working concertedly to make pyruvate turn into acetyl-CoA. So let's take a look at E1, E2, E3, and see what's going on there. All right, so pyruvate dehydrogenase multi-enzyme. This is still our step 0, right? We're not actually to the citric acid cycle yet. We're trying to get our pyruvate into acetyl-CoA. So step 0 of the citric acid cycle. So in this step, we're going to use our pyruvate that we just made in glycolysis. We're going to add a coenzyme A, and we're going to add 1 NAD+. From this, we should get the acetyl-CoA, which is ready, bless you, to head into the citric acid cycle. We're going to get 1 carbon dioxide molecule out and 1 NADH. Here's our pyruvate. Here's our CoA. I don't have that many more colors. Oh, there's black. So, the overall reaction requires all 3 enzymes. It is a 5-step reaction. So we're going to take a look at all 5 steps and see how they work before we get ready to actually enter the citric acid cycle. So the first enzyme, which we're showing here, E1, which is the pyruvate dehydrogenase, is an enzyme that requires TPP. We used TPP last week. Oh, before I forget, let me do this. Overall, this reaction is going to require 5 coenzymes that we're going to walk our way through. TPP, lipoamide, coenzyme A, FAD, and NAD+. So we've already talked quite a bit about CoA, FAD, and NAD+. We're going to spend some time looking at TPP and lipoamide. All right. So pyruvate dehydrogenase is a TPP requiring enzyme, which is one of our cofactors. One of the things it's going to do is going to decarboxylate pyruvate, which is how we get our carbon dioxide out of it. And it's going to make this kind of funky, weird intermediate, hydroxyethyl TPP-E1. So here's our TPP attached to our E1 enzyme, which is right here. So TPP in the active site of E1 in the presence of pyruvate is going to decarboxylate. Right, we're going to remove this carbon dioxide and form a new bond between this carbanion and what was the pyruvate. So that funky structure that has a carbanion and a protonated nitrogen is called a thiazolium ring. It acts like an electron sink, which means it really enjoys making carbon-carbon bonds, which means it's a great cofactor, right? It's going to make this carbon-carbon bond, and it hangs on to that full-on negative charge. That negative charge that was here is now down here. So it still has a carbanion, it's just moved from here to there. So it makes it a great cofactor, that it can make a carbon bond, but because it still has a negative charge, it's easily broken. You can make a carbon bond that then can be broken in our next step. So let's take our hydroxyethyl TPP E1 complex and head into step 2 with it, which uses our lipoamide cofactor. Yeah, we're here. So we enter pyruvate. Pyruvate kicks off a carbon dioxide and makes hydroxyethyl TPP. That then interacts with E2 and a lipoamide to make acetyl dihydroxylipoamide, which then is going to interact with CoA, kick off the acetyl-CoA, combine with this cofactor here to make E3 to then regenerate all the other ones and kick out MAD+. Yes.
Is that a 5 or an S?
5. 5. There's 5. 5.
This is all step 0.
This is all step 0. There's 8 in the citric acid cycle, plus step 0, which has 5. So really altogether 13. And then when we add in, eventually we'll get to adding in the electron transport cycle. We're not there yet. We just know the 10 of glycolysis to shuttle into the 13 of citric acid cycle. So it's an easy 23 simple steps. Step 0 is the hardest, for sure. It's the most complicated. The rest of them are much more straightforward. So we just made this, and we're going to interact it with lipoamide and E2. All right. So our hydroxyethyl group, which we just made over here, hydroxyethyl TPP-E1. Okay, so the hydroxyethyl that we just made on the previous slide, previous step, is now going to be transferred to enzyme E2. E2 is named dihydro. Dihydrolipoyl transferase. So dihydrolipoyl transferase. So it's going to transfer this lipoamide group. And this enzyme contains a lipoamide group, which is pictured right here. So the lipoamide contains a lipoic acid, which is this section right here. That's lipoic acid, attached to a lysine. And the chemistry that's going to do is right here. I guess let me do a different color. So in the presence of protons and electrons, we are going to break, what's it called when two sulfurs are linked together? Disulfide. So this breaks a disulfide bond to make two free ends. And then later on we'll regenerate it. So that's where the chemistry is happening. So the most, this part, as you can see in all the pictures, they just show it as like a squiggly line. This part, interesting, but not where the chemistry's at. So I'm not concerned about that end. I'm concerned that you can recognize this 5-membered disulfur ring. That's where the chemistry's at. So the reactive center, the lipoamide, is going to be that cyclic disulfide. So, in the active center, we have, let's zoom in a smidge bit. There we go. So here, bless you. There's our hydroxyethyl TPP-E1. This part right here, which was this guy right here. So it's now coming into the E2. And this full-on carbanion is going to attack one of the sulfurs and force the disulfide bridge to break. Then we're going to add 2 protons and sequence 1 to protonate this sulfur, and then the second one kicks off the original TPP-E1. And now we have this new intermediate. Yep. So all we've done is we've transferred the acetyl group. We had the acetyl group on the TPP, and now we have an acetyl group on the lipoamide cofactor. What it does? Okay, let me back up. Okay. So E2's job is to take what we just made, this hydroxyethyl TPP, and it's going to produce a free acetyl group. Not quite free. It's covalently attached. But we had it in this form, like an alcohol carboamide methyl group. And in the presence, the first proton protonates here. Then the second proton, best way to explain it, releases the TPP. And now we have the acetyl group, the free acetyl group. All right, so we're trying to get to acetyl-CoA. So we broke up the pyruvate. Now we have a acetyl, which is really what step 2 is doing. And another step or two will make the acetyl-CoA. So we're slowly breaking. Our first step is really to break it. Second step is to transfer it so it's now in acetyl form. Then we're going to CoA it, and then the last few steps are to get everybody back into their original shape.
So you said it was the hydroxyethyl into a free acetyl, or
Free is in, it's attached to E2. Right, but now that looks like an acetyl group, where before it was kind of a funky alcohol thing. All right, so then let's take this acetyl dihydrolipoamide, E2. Right, this is what we just made. Looking back at our entire picture. All right, so now we're here. We had this. Now we have this. Now we're looking to, there's little numbers here too. 1, 2. So we just did step 2. Now we're going to do step 3, which is adding the CoA and releasing the acetyl-CoA. So step 1, 2, 3 makes acetyl-CoA. Step 4, 5 regenerates the enzymes to their original. You might want to write that down. So steps 1 through 3, their job is to make the acetyl-CoA, send it on its way. Step 4 and 5 of this reaction will regenerate our enzymes so that they can do the whole cycle again. That's their specific purpose. So let's take a look at step 3, where we just made this acetyl dihydrolipoamide, and now we are looking to CoA it and send it on its way. So here's what we just made. So E2 catalyzes the trans-esterification reaction, which means it's going to transfer this group. All right, transferases, transfer groups. So the acetyl group is transferred to CoA. So CoA has a thiol on it, and it's stuck to a thiol. So we're just switching the acetyl group from one thiol-containing thing to another thiol-containing thing. At that point, we've now got our acetyl-CoA, and this is ready to head off to the actual citric acid cycle. Floats away, does some other chemistry. But now our E2, right, it was a cycle Now it's 2 thiols. So step 4 and 5, we're going to spend some time getting it back into a cycle so it can do another round of chemistry. So we need to regenerate E2 back to a cycle. So that's what we're going to use enzyme E3 for. So here's enzyme E3. Just a very, obviously it's much more than that, but here's the important part. It also has a cyclic thiol. And so it is going to do a disulfide interchange reaction. So we have a cycle, no cycle. Now we have no cycle, cycle. So it's going to transfer those electrons and protons so that E2 is now back to its original form. Yeah, I guess I already stated everything. So E3 gets reduced. It was oxidized, now it's reduced. Which means E2 was reduced and then it goes to oxidized. It's a redox reaction. So, are we done? We got one step left, right? Because we just reduced E3. In order for E3 to be able to come back and do this chemistry again, we're going to have to oxidize E3 back to its original shape, which is what the last step of this is. The oxidation of E3. So we're going to use NAD+, some free NAD+, that was from maybe glycolysis, or maybe later on the citric acid cycle. Use a free NAD+ to provide the electrons needed to go from the reduced E3 form back to our oxidized E3 form. And at this step, everybody's back to where they were. We have our TPP with nothing stuck to it. We got our E2 back in its cyclic form. And now we have our E3 back in its cyclic form. So here's my little cheat sheet of every of the enzymes, the cofactor, and what every enzyme does. So this should hopefully kind of clarify any of the points we may be confused on. So TPP is stuck to E1, and its job is to decarboxylate pyruvate. Lipoic acid is found in E2. It takes the hydroxyethyl carbanion from E1 and makes it an acetyl group. CoA, then it takes the acetyl group, releases it as a CoA. Then FAD and NAD+ are in E3 to do some oxidation reduction chemistry and get everybody back to their original shape. Questions about step 0? In that case, let's just do a little overview of the enzymes in the citric acid cycle. Everybody looks shell-shocked because it was just so amazing how that worked. How simple, regenerative.
How fast will the cycles go?
Super, super fast. I mean, because that's a 60 enzyme complex, it's able to turn over thousands of pyruvates into acetyl-CoA in seconds. It is right at the rate of diffusion. So it's as catalytically perfect as it can get, which is good because your body needs because obviously there's more than one citric acid cycle going at a time. When we think about it, when we're discussing it, it may seem like there's one enzyme of each per cell. That's not right. There's many of each of these enzymes per cell, so your body is able to do multiple glycolysis citric acid cycles in one cell, and then all your cells are doing them, some more than others, right? So just about catalytically perfect. Then we'll get a little bit into brain chemistry later, because your brain needs other things, because your brain runs mostly on glucose, so it has some different needs than the rest of your body. So, which is why you should eat a lot of sugar while studying, because your brain really needs a lot of glucose, because it's its only method of getting ATP. So I think that's my method of studying. Heavy on the Halloween candy. All right, so I'm just going to write down for each one of these enzymes what their specific job is, and then Friday we'll look at each one of them and see how they do it, what their energy needs are, what their intermediate, if they or cofactors, if they need it, will be. So citrate synthase is considered the first reaction of the cycle, and it's going to take the acetyl-CoA and condensate it. What kind of molecule do you need when you do a condensation? You're going to need some water. So be thinking that we're going to need some water. And we're going to condensate it to oxaloacetate. I've talked to any of these guys before. Guess I haven't. Okay, so then we're going to do a reversible isomerization, which, if you had to guess, what would the delta G of the reversible isomerization be? 0. It should be 0, so this should be a 0 step. Good. Go. All right, so then isocitrate dehydrogenase does a decarboxylation of isocitrate to alpha-ketoglutarate. Any guesses on what one of the products will be besides alpha-ketoglutarate if we do a decarboxylation? Carbon dioxide. And if it's oxidative, what sort of reaction should this be? Redox reaction. So we're also going to make an NADH. Let's zoom out. Okay. So then we're going to take, that should be an alpha, not an A, alpha-ketoglutarate. Alpha-ketoglutarate dehydrogenase is then going to catalyze it's going to catalyze another oxidative decarboxylation to make succinyl-CoA. So what would be some expected products from a catalytic oxidative decarboxylation? And then we're doing another decarboxylation, so yep, we get our other CO2 at this step. Do we make NADH? I do not think we, no, we make all NAD. Yeah, so, okay. Got confused. All right, so then, now we've made succinyl-CoA, we're gonna react succinyl-CoA with succinyl-CoA synthase. So we're going to cleave our high-energy succinyl-CoA into succinate and CoA. So if we are cleaving a high-energy bond, what is most likely going to be made at this step? ATP, but we're in the citric acid cycle, so it's GTP. So this step is where we make our molecule of energy. Succinate dehydrogenase, as its name implies, catalyzes a dehydrogenation reaction, but it's funky because it's stereospecific dehydrogenation. So we're going to make something chiral at this point. We're going to make what's called fumarate. Lucky for us, the next enzyme is fumarase. So I do like the citric acid cycle because the intermediates are named for the enzyme that uses them next, in general, right? So isocitrate uses isocitrate dehydrogenase, which then makes alpha-ketoglutarate, which then uses alpha-ketoglutarate dehydrogenase. So the intermediate and the enzyme that uses it have the same name. So that should help with the learning. So then our last two steps is we're going to hydrate a double bond of fumarate to make malate. Then malate dehydrogenases regenerates our original molecule. So we get back our acetyl-CoA from the top, and then we'll start back across. So, Friday, you will have a guest lecturer